Now in 513, we are looking at two female Drosophila. Each of them are heterozygous for a couple different genes here. So we have B for black body, recessive here. D, put this mic a little closer, sorry. Okay, D for Dox tarsus, Dox, and C for curved wings. Now I don't work with Drosophila, so I'm not exactly sure how to, pronounce something like Dox tarsus. And not sure what it is, but it doesn't matter. All the standard concepts are the same between organisms. So C is for curved wings. Now the genes are in the order DBC, so I can draw a little map of a chromosome D, B, C, with B closer to D than to C. Okay, so that's the setup to the question. Now let's see what the main question is here. So we have one female. Let's go with female A. Now she has the following genotype with arrangement of alleles. So D, B plus. So remember, when we talk about arrangement, we mean how the alleles are arranged on the chromosome. So these D, B, and the wild type C allele, this plus here, is they're all linked on one chromosome, and the wild type D, the wild type B, and the mutant C are all linked together on the homologous chromosome. So we're using that shortcut method where instead of writing out D plus, B plus, C plus, we're just using pluses. And we can do that when we write the genotypes like this, with a plus right underneath the D, a plus under the B, and a plus over the C. And we know the order of the genes, so we know that every time we see a plus in this first position, it has to be a D. A plus in the middle position, it's a wild type B allele, and a plus in the last position is a wild type C allele. So essentially, the problem lists out the eight possible genotypes that can result from gametogenesis in this female, and they number them 1 through 8, and we're supposed to decide which ones are the non-crossover gametes, which are the single crossover gametes, and which are the double crossover gametes. So I'm just going to write those out for you, not the way they're listed in the problem, but let's just figure this out without even looking at the problem. So this female right here, meiosis in this female, is going to produce two types of gametes, non-crossover gametes, right? So one of them is going to be DB plus, and you know what, I'm not going to put the 1 there, just one is going to be DB plus, so one gamete here, and the other one is going to be plus C. So if no crossing over occurs during meiosis between D, B, and C, well, these are the two gametes that we're going to get. Gametes. And these are the non-crossover gametes. And one of the things we should point out is that, these gametes are making up one reciprocal class, and they're reciprocals because everywhere we see a D, we see the opposite, plus. Everywhere we see a B, we see the opposite allele, so B plus. And here, every time we see a plus up here, we see a C down here. So these are reciprocal genotypes of each other, and these two genotypes make up one reciprocal class, and in this case, this is the non-crossover class. So now let's look at what happens when we get a single crossover at this position right here. So what we need to do is move this C up here and this plus down here. So when we move the C up here, move the C up, we end up with the genotype D, B, C, and plus, plus, plus. So that is one single crossover class of gametes, and that resulted from a crossing over at that position. And you can see, you know, these gametes make up one reciprocal class, and they are reciprocals of another. So one gamete has every mutant allele, and the other gamete has every wild-type version of that allele. So when we have 3 genes and we're looking at single crossovers, one class is going to be made up of a single crossover between 2 of those genes, and the other class is going to be a single crossover between the other 2. So let me move this down here. So now we want to look at the case where we have a crossover here. And one thing the way I like to do it, I like to take both of these now and move both of those up top, and then take 2 of these here and move both of those down. And why do I like to do that? I like to think of, you know, if the crossover is happening here, and then we're physically exchanging both of these things, and both of those are moving together, and then both of these are moving down. So the other way to look at it is, okay, we can have a crossover here, well then we can just move this one up and this one down. So either way you want to do it is fine. So following either one of those approaches, we are going to have, let's see, D, and I'm going to move plus and C up here. So D plus and C in one gamete, and plus, now moving B and C down, right, because the crossover is happening here. So B and plus are moving down. And now we have the other reciprocal class of single crossover gametes. So there's 2 classes. I'm going to name this one 1. Doesn't matter which one you name 1 or 2. I'm going to name this one 2. And you can see that these are reciprocals. These gametes are reciprocals of one another. I wanted yellow there. Everywhere we see a wild-type allele in one of the gametes, we see the mutant allele in the other, so they're reciprocals. Okay, so we have one more class to talk about, and that is the double crossover class, so the DCO class. Now for that, the way I like to look at it is, and I do things the long way, I don't know why I do, I just do. I'll show you the shortcut way next. But what I like to do is I like to take these here. I like to assume a crossover happens here first. This plus and C, well, that'll move up top here, and the B and plus will move down. So I could put the plus and C up here, and the B and plus down here. And then I like to assume the crossover now happens there. So first it happened over here, second it happens here, and because of that I gotta move this C down and that plus up. So the end result is D, B, C, right? I don't think that worked. Why didn't that work? Because the plus is moving, oh no, D plus plus and plus B, C. Okay, now that's the way I like to do it, and you can see if you're not careful, you're prone to mistakes. But you know, knowing how to do that may help you solve some of the more complex genetic problems, because I think it helps you see things a little better. Okay, the shortcut way though, if you're ever in doubt, just take this middle allele and switch that. So B goes down, plus goes up. So let me map that out here. So B is going to go down, plus is going to go up. So we'll have D plus plus and plus BC. Now that's the double crossover class of reciprocal gametes. Or reciprocal class of double crossover gametes. Sorry. So, okay, so we have all 8, and then what we would need to do is go back to the problem and just match these up. And if I'm looking at the problem right now, and you can look in your books, or if you have the e-text, look on the e-text, and you'll see that these are numbered 5 and 6. These are numbered D plus C 7 and 8. These are 1 and 2, and these are 3 and 4. Okay, so the next part of this question, before looking at female B, was to determine the relative order of the gametes resulting, well, in this female right here. So to answer this, what we need to do is know that what we need to remember is that no crossover gametes, non-crossover gametes, are always going to be found in the highest level. And that has to do with the fact that in every time you have a crossover, right, in a tetrad, well, you have at least 2 of the chromatids are not, 2 non-scissor chromatids aren't going to be involved in that crossover. So at least 50% of the gametes are going to be non-crossovers, and no gamete is going to be higher than 50%. So non-crossovers are always going to be found in the highest level. And we learned from question, I think it was 5.6 or 5.7 or somewhere around there, that double crossovers are always less frequently encountered than the single crossovers that make up the double crossover. So those are the least frequent. So in this case, 3 and 4 were the non-crossovers, so they're going to be found in the highest, at the highest frequency in the gametes. Double crossovers, those were 5 and 6, so they're going to be found at in the least amount. But those single crossovers, now that's where the trick is here. So are 1 and 2 going to be found more often than 7 and 8, or less often? So we have to go back to that map. Remember, the question told us that B was closer to D than to C. And because we know crossovers happen randomly along the chromosome, because of that, they're more likely to happen between B and C than they are between D and B. So we're more likely to see crossing over between these two than we are between these two. So we need to find the single crossover class that resulted from a crossover between B and C, and that's going to be found, those gametes resulting from that cross are going to be found more often than the gametes resulting from a crossover between D and B. And so let's look at the way I map this out. I want the crossovers between B and C because they're going to be found at the highest amount, and that's this class right here, what I called single crossover 1. You can see if this is the arrangement of alleles in the mom here, this one moved up, this plus moved down because of the crossover here. We have DBC together and plus 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 together. So these are going to be found more frequently than these gametes in this single crossover class here, because B and C are further apart than D and B. So 7 and 8 are going to be found single crossover class 1, is going to be found more often than gametes in single crossover class 2. And that was, oh wait a second, was it 7 and 8? Let's see. No, 7 and 8 was 2. And these were 1 and 2. Okay, so this is the relative order of the gametes that we should see from that female. And again, so this one was easy. Non-crossovers always found most frequently. Double crossovers always least frequently. And single crossover class 1 was found more frequently because B and C are further apart than B and D. And because they're further apart, we're more likely to see single crossovers between those two genes than we are between these two genes. I'm not going to do female B because it's exactly the same as female A, so you should be able to figure that out on your own. Okay, I'm going to move on to the next problem.